All right, we have finished the metabolism handbook. Now we are going to go back to the case. Claire's symptoms suggested there was a problem with her metabolism. Let's look at Claire's health. To help restore homeostasis in Claire's body, you first need to know more about the metabolism. This is where we went through the handbook. Let's investigate Claire's weight loss. Claire is eating a lot of food, but is still losing weight. This suggests that food is not being broken down by Claire's enzymes. The process of breaking down food by enzymes is called digestion. Let's see what is in food. Food contains many nutrients, including proteins, carbohydrates, and lipids, or fats. During digestion, enzymes break the nutrients down into small molecules that the body can absorb or take in. We are now going to move on to our lab data. Let's look at our digestion. We are going to feed Claire. As the food goes down to her stomach, let's go inside and see the digestion. In the stomach, proteins in food are broken down by an enzyme called pepsin. Pepsin is released by the cells in the lining of the stomach and work best at a low or acidic pH. The stomach maintains the low pH that pepsin needs by releasing hydrochloric acid. Let's follow where it goes after we go through the stomach. So from the stomach, the food goes to the small intestine. Let's see what happens to the food here. Enzymes in the small intestine break down the proteins for carbohydrates and lipids. These enzymes are named after the substrate they break down. The protease enzyme breaks down proteins. The carbohydrates enzyme breaks down carbohydrates. And the lipase enzyme breaks down lipids. Let's review. The enzymes in the stomach were pepsin. It breaks down proteins and requires a low pH. We then have in the small intestine protease that breaks down proteins, lipase that breaks down lipids, and carbohydrates that breaks down carbohydrates. Now, let's go ahead and collect some data. First, we have to collect some data from a healthy dog. To do this, we will take 10 milliliters of liquid from a healthy dog's stomach and 10 milliliters from its small intestine. Next, we will add a milliliter of the liquid from the stomach of the healthy dog into a solution of 1 milligram per milliliter of protein, the substrate, to see how it does for digesting the protein. So we want to pick up this blue dot and drag it over to the protein solution. Now let's add our data to a graph. Here are our stomach proteins. This is how much substrate's there. The graph shows how the protein concentration changes over time when the stomach liquid is added to the protein. As the enzyme breaks down the substrate concentration should go down, as we see in the healthy dog. The data from the healthy dog is our control data. We compare this data to the data from Claire. Now let's run the same experiment with a milliliter of liquid from Claire's stomach. Let's collect Claire's data and add it to the graph. Now you should notice that Claire's line and the healthy dog's line are in the same spot. Let's analyze our data. Here we have our question. Compared to the healthy dog, did the enzymes from Claire's stomach break down the protein at a low, normal, or high rate? Well, both the lines were at the same spot, and the yellow line was from the healthy dog, so we could say that was normal. The data shows the protein was broken down at the same rate in both dogs. Now we need to examine the enzyme activity in the small intestine. The way we'll do this is we'll take a milliliter of the liquid from their small intestines and put it into the solution of either protein, lipids, and carbohydrates. Let's collect that data. Remember, the enzyme breaks down the substrate, so the substrate concentration should be going down. Did the enzymes from Claire's small intestine break down the protein at a low, normal, or high rate? Well, when we look, Claire's line, the red line, is not using up that substrate nearly as quickly. So it is doing both the protein, lipids, and our carbohydrates at a very low rate. The data shows the enzymes from Claire's small intestine broke down the nutrients much slower than the enzymes from the healthy dog. So where was the problem in Claire's digestion? We want to make a decision here and hit submit. We can then go on to our diagnosis. There are three possible causes of Claire's problem. Let's look at our first possibility. The first possibility is the enzyme active site mutation. Here is what a normal active site, and this is a mutated active site. Sometimes when a cell makes an enzyme, one of the amino acids can be changed to a different amino acid. This is called a mutation, and it can change the shape of an enzyme's active site. This could stop the enzyme from breaking down food molecules. Let's check our next possibility. An enzyme's shape depends on the pH activity of its environment. If Claire's body was not keeping her small intestine at the correct pH, none of her enzymes would work properly. Finally, let's take a look at the possibility of low enzyme production. For low enzyme production, an enzyme can only act on one molecule at a time. This means the reaction rate depends on the concentration of enzyme. If Claire makes enzymes too slowly, the enzyme concentration in her small intestine would be low. A low concentration would cause a low reaction rate and food would be broken down too slowly. Let's perform two more experiments to make our hypothesis. Let's repeat the experiment to measure the breakdown of protein by liquid from the small intestine. This time, we'll take the 1 milliliter of liquid and we will add it to the substrate, but we will do it at a pH of 9, which is the normal pH of the small intestine. The graph will show the breakdown of the protein. Let's start with the control data from the healthy dog. Here's our healthy dog's line, and let's collect Claire's data. Here's Claire's line. So both Claire's and the healthy dog's liquids were placed in a pH of 9, so they should have had the same pH, and here Claire is still having lower function. We have taken 1 milligram of pure protease out of the small intestine liquid. So instead of just taking a milliliter, they sifted out 1 full milligram of pure protease from the small intestine liquid. And then we added that protease from the healthy dog to the substrate. And again, it'll show the rate of protein breakdown by milligram. So let's collect our control. Here's our control. It looks very similar to this one. Now let's do the same for Claire's. So when we take Claire's enzymes out, and we have the exact same amount of enzyme for both the healthy dog and Claire, their graphs line up again. Well, let's go ahead and make our hypothesis. Using the data, we want to choose one of our three possible problems. Once we have done this, we do want to justify. And for this first time, the way we are going to justify is you are going to explain, I think it is choice A, B, or C. You want to make that choice because, and here you want to use data from the graphs above. Once you do this, you can click Submit. Now we're going to go ahead and test our hypothesis. To test our hypothesis, we are going to perform 3 experiments, 1 experiment for each possible enzyme problem. Let's start our first experiment for mutations. Let's examine the binding of substrates to the active sites of Claire's enzyme. First, we need to collect a sample to get a close look at each enzyme. Now we need to analyze this sample. Here are the 3 enzymes in the small intestine. Each enzyme's active site is shown with a yellow highlight. and here are their substrates. Now we want to drag and drop to match which substrate goes with which, and click Submit. Then we can bind the substrates to, and let's see our results. All of these have 100% shape match. So let's go ahead and revise our hypothesis. So now with this piece of data, we may want to revise our hypothesis because since we have 100% shape match, enzyme active site mutation is no longer a possibility. So we want to add the active site mutation is not correct because, and here we want to use the data from above right here in order to do this. And then we can make a new hypothesis and hit Submit. Let's continue on to our next experiment, the pH range. Enzymes work best within a specific pH range. This is called the optimal or normal pH range. If Claire's small intestine has an abnormal pH, her enzymes would not break down food fast enough. To test the effect of pH on Claire's enzymes, you need to examine the reaction rates on her enzymes at different pH levels. In the optimal range, the enzyme successfully breaks down food. If I slide this to low, our enzyme is failing to break down food. Now, if I slide it up to high, the enzyme is again failing to break down food. Let's collect some reaction rates from a healthy dog. We want to begin by investigating the reaction rate of Claire's protease enzyme at pH of 6. To do this, we will add 1 milligram of protease from Claire to a milligram of protein substrate at the pH of 6. The amount of product made will be on the y-axis, and time will be on the x. When we add the milligram of protease and collect our data, we find our reaction rate equals the product made and time. So the protease made 1.5 milligrams of product in 3 seconds. What was the reaction rate? We would take 1.5 and divide it by 3. We can now look at how the reaction rate changes with pH. At a pH of 6, the reaction rate was 0.5 milligrams per second. We'll plot this on a second graph. Our reaction rate will be on the y-axis and pH on the x-axis. Which dot shows a reaction rate of 0.5 at pH of 6? Well, let's start off by looking at our pH of 6 and going up. The only dot that has 0.5 as the reaction rate and 6 for the pH is this dot here. We are now going to analyze our data. Let's measure the reaction rates for carbohydrates and lipase. These graphs show the reaction rates of protease, carbohydrates, and lipase at various pH levels. We can now determine the optimal pH range of each enzyme. This is the pH range at which an enzyme works at 90% of its maximum reaction rate or more. Let's highlight our optimal pH range. Next, we'll measure the pH in Claire's small intestine. It is 9.1. We want to see if the pH of Claire's small intestine is within or above the optimal range of each enzyme as we look at these graphs. You want to answer these and click Submit. We can then revise our hypothesis again. Now here, we can identify that abnormal pH is not correct because, and again, use data from above and click Submit.